Hello everyone, I will try to explain saltatory conduction in this video session. What is exactly this type of conduction? And what is the advantage of having saltatory conduction? So conduction is nothing but the transmission of action potential from one particular side of the neuron to the other side. So that is conduction of action potential, transmission of action potential. We all know that in our body we have two types of nerves. The one is called as the cranial nerves. The other one is called as the spinal nerves. Cranial nerves are arising from the brain and spinal nerves are from the spinal cord. Whatever it is, these types of nerves belongs to peripheral nervous system. Okay, you take the peripheral nervous system nerve. Okay, so that's what you see here. I have drawn a small portion of the nerve here. Both are peripheral nerves itself. That is one could be a cranial nerve and the other could be spinal nerve, whatever. Okay, so we know that dendrite and axon and cell body are the main parts of the neuron. Here I have drawn the axon, a portion of axon here. So you could see two diagrams, that is nothing but axon. We all know that there is a specialized cell called as the glial cells which actually present around the axon plasma membrane. So these rectangular boxes are nothing but the glial cells. Specifically, they are called as Schwann cells. So in human neuron, you see Schwann cells which is actually wrapped around the plasma membrane. Okay, they have specialized function in our body, that is they can produce a fatty layer around the plasma membrane of the Schwann cell. Okay, so fatty layer will be produced, that is actually colored in dark. Okay, so like this around their plasma membrane, and they are called as myelin sheath. That's it. The fatty layer which is produced by the Schwann cell is called as myelin sheath. If the Schwann cell which is present around the plasma membrane can produce myelin sheath or fatty layer, then that type of axon is called as myelinated axon. Okay, so we just put D here. And if the Schwann cell are unable to produce that myelin sheath, that is fatty layer around the plasma membrane of them, then it is called as non-myelinated axon. That is only the difference. But there is one advantage in myelinated axon, that is when action potential, so I just here write action potential which is now generated in the dendrite of this particular myelinated neuron, okay, which now arrives to the axon hillock. This is the starting point of the axon, which is actually called as axon hillock region. And here you could see the plasma membrane which is wrapped by the Schwann cell which is actually having the myelinated sheath, actually myelin sheath. Okay, so here we have action potential. But now the interesting thing is here, that is between the two Schwann cells, there is a gap. This is the gap actually, and the gap is actually called as, we'll write here, nodes of Ranvier. So wherever the nodes of Ranviers are present, there you see extracellular fluid. That means this particular axon is exposed to the outside. That is, this is the region where insulation is not there. That is actually nodes of Ranvier, where ECF is there. What is ECF? Extracellular fluid. Okay, so this is the site of the axon which is now exposed to ECF. Okay, and here you could see the sodium and potassium ion channels. High concentration of sodium and potassium ion channels present only in the nodes of Ranvier, but even we have sodium and potassium ion channels here within the region where the insulation is there, that is by myelin sheath, but very less number. Okay, here we have more or high concentration of sodium ion channel. They are voltage gated channels. Okay, now here we all know that for the action potential to generate, we need reversal of the charge. We know that here when the axon plasma membrane is not conducting any action potential, the inside is always negatively charged, outside, that is ECF, is positively charged. So for the generation of action potential, we need to reverse, that is positive charge should come inside the axon and negative charge should go. Okay, so then only action potential will be generated. But for the generation of this action potential, for the reversal of the charge within the or across the plasma membrane, we need rapid movement of the sodium ions towards the inside from outside. So sodium ions, they are present in the ECF, should get inside, okay, for the reversal of the charge to generate action potential. But here this particular region is highly insulated, highly insulated where there are no sodium, well, like very less number of sodium ion channels, and as a result sodium cannot get inside, okay, to depolarize this region. We have only here the sodium ion channels are present, okay, in the nodes of Ranvier. So action potential should skip this region where insulation is present. So skipping of action potential where insulation is very, very high, okay, in the region of axon. So now this action potential jumps to the site where sodium ion channel as well as potassium ion channels are present, okay. So this is ECF. So this will be seen where insulation is poor. So action potential will be generated only in the region where insulation is not there, myelin sheaths are not there, that is in the nodes of Ranvier, okay. So this is really advantage because skipping of action potential will normally takes very less time. Ultimately, due to this, the movement of action potential, that is transmission of action potential, will take very less time, that is it is very, very fast within the axon. If you take non-myelinated axon, here we don't have any myelin sheath, there is insulation is very, very poor, actually there is no insulation, and ultimately I just want to tell you this, that we have sodium and potassium ion channels throughout the plasma membrane of the axon, like this we have it. And now action potential which arrives from the site of the neuron, that is dendrite, and here at each site wherever these ion channels are there, depolarization should take place. So normally it takes a lot of time. At each site depolarization takes place, that is reversal of the charge across the plasma membrane, that is electrical as well as chemical potential difference across the plasma membrane should occur throughout, because we have all sodium and potassium ions throughout, and at the same time there is no insulation. So this particular plasma membrane is exposed to ECF, there is no insulation, and ultimately this is time consuming process, that's it. So this is actually saltatory conduction.